Hello everyone, today we'll talk about the chapter Molecular Basis of Inheritance. So first of all, before starting this chapter, I would like to tell you the meaning of this chapter. So according to this title, what are the molecules which are being transferred in the process of inheritance? Now inheritance means the characters that you get from your parents. So whatever characteristics you are getting from the parents, for example, hair color, eye color, height, weight, whatever it is from the parents. So this is your inheritance. So what are the molecules which are being transferred in the process of inheritance? We call this molecular basis of inheritance. So in human beings, what we get from our parents, it is the molecule DNA. DNA stands for deoxyribonucleic acid. In this the DNA, if we talk about its structure, DNA is double-stranded. DNA is double-stranded. It means it has two strands. It has two chains. Now these two strands are called nucleotide chains. These two are called nucleotide chains. These nucleotide chains, if we talk about its structure, what are these chains? These are not just two strands, just like a thread. These are actually contains some chemical structure. So if we talk about its chemical structure, it contains nitrogenous base. It contains a pentose sugar of carbohydrate, and it contains a phosphate group. This is all about nucleotide chains. If you don't understand the nucleotide base or pentose sugar or phosphate group, now let me draw a structure of one nucleotide chain of DNA so that you can understand it in a better way. So we'll talk about the nucleotide chains now, which is a part of DNA. So let me draw you a structure so that you can understand it in a better way. This ring. It is a pentose sugar. Pentose means the sugar which has 5 carbons. So 1, 2, 3, 4. Here we have 4 carbons. Now what? Now here we have the 5th carbon. This is the 5th carbon. On the 3rd carbon we have one OH group which is attached here. Now I talk about the third thing, which is the phosphate group. Now let's talk about phosphate. Where is this phosphate group? Here is your phosphate group. This is the structure of DNA. Now if you can see, on the 5th carbon, 1, 2, 3, 4 and 5th carbon, we have phosphate group attached. So we call this group as 5' phosphate group. We call this as 5' phosphate group, or simply we call it as 5'. And on the 3rd carbon here, we have an OH group attached. So we'll call it as 3' OH group, or simply 3' end of the nucleotide chain. So as we know that DNA is double-stranded structure. So how the other strand would be? This is one strand. Now other strand would be, their phosphate group shall be on this side and the OH group shall be on this side. So this chain would be exact antipolar of this current chain. The second chain would be the complementary. It shall be the complementary and antipolar to each other. Now let's talk about the very third aspect of this nucleotide chain, which is the nucleotide base. Now we'll be talking about this nucleotide base. What are these bases? Nucleotide base or the nitrogenous bases. It is also known as nitrogenous bases. So these bases, these are nothing but these are the very essential parts of the DNA which connects these two chains. For example, how are these chains are connected to each other? Why they are not separating? So these are the bases which are connecting these two chains. Now let's talk about these nitrogenous bases. So first of all, we have two types of nitrogenous bases. First is purines and second is pyrimidines. In purines, we have two types of bases, adenine and guanine. In pyrimidines, we have three types of bases, thymine and cytosine. And the last is uracil. These are the three bases that we have here. But now look at this. But DNA has only these four kinds of bases, adenine, guanine, thymine and cytosine. Uracil is present in RNA only. It is in RNA only. It is not present in the DNA. Now let's talk about these four bases. We'll call this as A, T, G and C in short form. So adenine will make double hydrogen bonds with thymine and guanine will make triple hydrogen bonds with cytosine. So this is how they make bonds in the DNA. Now let's talk about these two, how these are connecting with each other. So from one nucleotide chain, if we have A, adenine, so other base will have thymine because it is a complementary base for the adenine. A won't be connected to each other. Adenine would not make bonds with each other. Now let's talk about the cytosine. If we have cytosine, it will make triple bonds with the other chain by using guanine. So this is how these two chains are connected with each other. Right? Now let's talk about a few features of DNA. First of all, Watson and Crick were two scientists who gave the structure of DNA by using a technique which is called X-ray diffraction. X-ray diffraction in 1953. So these were the scientists who gave us the structure of DNA. And there was one more scientist. This is the first feature of DNA. Second feature is there was a Chargaff scientist. This is the name of the scientist who gave us Chargaff's rule. So as we have already studied these four bases of DNA, what happened? Adenine plus guanine ratio and thymine plus cytosine ratio shall always be 1. This is what the Chargaff rule said to us. This is what Chargaff rule stated. So how this thing is possible? So first of all, look at this very carefully. Whatever adenine we have thymine shall be equal to it because they always make double bond with each other. Again, guanine will always be making bonds with the cytosine. So here they'll make triple bonds. So that means this and this number is equal and guanine and cytosine are equal. So that is how the ratio is 1 because their number is also equal in number. Now third feature of DNA is the two nucleotide chains of DNA. These are anti-parallel and antipolar. Now what are these two terms, anti-parallel and antipolar? Now if you remember, I said one chain shall be from 5' to 3' and the other chain would be in opposite direction. That means 3' would be here and 5' would be here. Now if you can recall, 5' was the phosphate group and 3' was the OH group. Again, 3' shall be OH group of the other chain and 5' shall be phosphate group here. So this is how they are anti-parallel and antipolar. Right? Now the fourth feature of DNA is between these two base pairs as in the adenine and thymine, guanine and cytosine. The distance between these two base pairs is 0.34 nanometers. So in one turn of DNA, 1 turn as in this turn. In 1 turn, we have approximately 10 base pairs, right? So in one turn of double helix structure that we have of DNA, the distance shall be, if you multiply these two things, 3.4 nanometer. If we talk about the DNA coiling, the DNA coiling is in right-handed fashion. Right-handed fashion. This is all about the DNA. Now there is one more term which is called central dogma. There is one more term which is central dogma. This tells us how the information flows in our system, right? So first of all, we have DNA, which we inherit from our parents. DNA's information is being transferred into mRNA. M stands here for messenger RNA. Messenger means something, a messenger transfers the information from one form to the another, right? From mRNA, we can make proteins. From mRNA, we can make proteins. This is what the central dogma states. Now, let's talk about the process which are being involved here. So, DNA can replicate itself. DNA can make copies of itself by a process called replication. When the DNA makes mRNA, messenger RNA, we call that process as transcription, because the information is being transcribed here in the form of mRNA. Now, making of proteins from the mRNA, we call this process as translation. This is what central dogma states. But now there is one reversal process which is available that the scientists have studied, that there are a few viruses which can make DNA by using mRNA. They can make DNA by using mRNA, like information can flow from mRNA to DNA. This process is called reverse transcription. Reverse transcription. And for this, the viruses needs an enzyme. That enzyme is called reverse transcriptase.